dear students, today we are going to discuss mechanisms of steroid hormone action. The main objectives of today's lecture are to get a concise thought about the steroid hormones, followed by highlighting the types of hormone receptors. Moreover, we shall try to know about the steroid hormone function. And finally, we shall try to understand the mechanism of steroid hormone action and its effect in detail. Let's first talk about the steroid hormones. Hormones like adrenocortical hormones and sex hormones are synthesized from cholesterol in several endocrine tissues. Such hormones are lipophilic and can diffuse the plasma membrane while they travel to their target tissues through blood circulation, bound to carrier proteins interacting with the intracellular receptors. The chemical structure of steroid hormones is similar to that of cholesterol, and in most instances they are synthesized from the cholesterol itself. They are lipid soluble and consist of 3 cyclohexyl rings and 1 cyclopentyl ring combined in a single structure as shown in the figure. Much of the cholesterol in steroid producing cells comes from the plasma, but there is also a de novo synthesis of cholesterol in steroid producing cells. Since these steroids are highly lipid soluble, once they are synthesized, they simply diffuse across the cell membrane and enter the interstitial fluid and then the blood circulation. All steroid hormones act through nuclear receptors to change the level of expression of specific genes, and the receptor hormone complex then translocate to the nucleus and interacts with specific response elements, stimulating the transcription. Well, dear students, now you might be well aware that steroid hormones are synthesized from cholesterol. Moving ahead, we will have a short discussion on types of hormone receptors. Hormone receptors are large proteins, and each cell type has its own combination of hormone receptors, which define the range of its hormone responsiveness. Also, the receptors are highly specific, so all hormones act specifically in hormone-sensitive target cells, to which the hormones bind with high affinity. Hormone action requires binding of the hormone to a receptor at the target cell. This allows the hormone to be distinguished from all other substances and to activate a cellular response upon hormone binding. A target cell responds to a hormone because it bears receptors for that particular hormone, and such binding of hormone with its receptor alter cell activity either by stimulating or inhibiting a number of characteristic cellular processes of their target cells. Further, regulation is achieved by a variable number of hormone receptors per type of target cell. Hormones can be grouped into 2 categories according to the location of its receptor at the target cell, either on the cell surface receptors, or intracellular at the level of the nucleus, nuclear receptors. Most peptide hormones bind to cell surface receptors, while the amino acid derivatives and steroid hormones are usually ligands for nuclear receptors. In case of any mutational change in the structure of a hormone receptor, it may result in a constitutive inactivation or activation of the hormone binding, leading to clinical scenario of the hormone deficiency and excess, respectively. Now, cell surface receptors. Cell surface receptors are glycoproteins that are highly mobile within the plasma membrane. Hydrophilic portions of the receptor are exposed at the cell surface, while the hydrophobic portions of the molecule are buried within the lipid bilayer. Cell surface receptors bind water-soluble hormones such as peptide hormones, monoamines and prostaglandins. Since these water-soluble hormones are not able to traverse the lipid bilayer to enter the cell surface receptor serves to transmit the hormonal message to the interior of the cell. The binding of the hormone to the cell surface receptor is reversible, allowing the receptor to be activated repeatedly. Although the hormone receptor complex may also be internalized, thus producing a single response from a single ligand receptor interaction. Although there may be a variable number of cell surface receptors, the principal target tissues for a particular hormone generally contains the largest complement of receptor molecules and are exposed to the highest concentration of the hormone. The binding of a hormone to a cell surface receptor stimulates a cascade of complex events through the generation of second messengers. The activation of protein kinases results in phosphorylations and altered conformation of a diverse number of molecules, which then produces a series of metabolic effects. Post-translational modifications of the receptor can affect downstream signaling pathways. Pathologic factors such as genetics, autoimmune processes and exogenous toxin exposures may further contribute towards the regulation of hormone sensitivity. Cell surface receptors which trigger intracellular signaling pathways can be subcategorized by their different molecular mechanisms: ligand-gated ion channels, receptor tyrosine kinases, receptor serine threonine kinases, receptor guanylate cyclase, G-protein coupled receptors, and cytokine receptors as well. The action of G-protein coupled receptors and cytokine receptors depends on the recruitment of other molecules, while the remaining types of cell surface receptors can also function as ion channels or enzymes to achieve effector function. Thus, hormones may utilize a variety of intracellular mediators, and a given hormone may utilize one or more of these intracellular pathways. The metabolic events regulated by the activation of cell surface receptors may either be rapid alterations in ion or substrate flux across the plasma membrane, or slower alterations in protein levels by modulation of gene transcription. Dear students, now we will discuss nuclear receptors. Nuclear receptors are a family of highly conserved transcription factors that regulate transcription in response to small lipophilic signal molecules. They are ubiquitous and unique to animal kingdom. Members of nuclear receptor superfamily can both positively and negatively regulate transcription. Nuclear receptor superfamily consists of an N-terminal activation domain, a central DNA binding domain, and a C-terminal ligand binding domain. N-terminal domain contains an AF1 domain, that's activation function 1 sequence, which functions as a ligand-independent transcriptional activator. AF1 domain is recognized by either coactivators or other transcription factors. Nuclear receptors act as regulatory transcription factors and bind with DNA regulatory sequences termed as a response element. Lipid-soluble hormones are small ligands and thus able to penetrate the plasma membrane to interact with intracellular nuclear receptors, which are much larger proteins. The classic nuclear receptors are those for the thyroid and steroid hormones, the latter which include aldosterone, cortisol, estradiol, progesterone, and testosterone. Vitamin A and D metabolites are other lipophilic signaling molecules that also utilize nuclear receptors. Nuclear receptors bind to lipophilic ligands with high affinity. Specificity of ligand receptor binding is accomplished by binding of the nuclear receptors to the hormone response element. That's the gene sequences of the ligand. Most nuclear receptors bind to hormone response elements as dimers, as the metabolic effects of these proteins are then produced by the translation products of the thyroid or steroid hormone regulated mRNAs. The action of these hormones are relatively slow compared to cell surface receptors. Dear students, by now we understood nuclear receptors. Now we will be discussing steroid hormone functions. The steroid hormones secreted by the adrenal cortex, ovaries and testes are to cause synthesis of proteins in the target cells. These proteins then function as enzymes, transport proteins or structural proteins which in turn provide other functions of the cells. The sequence of events in steroid function is essentially the following. The steroid hormone diffuses across the cell membrane and enters the cytoplasm of the cell where it binds with a specific receptor protein. The combined receptor protein and hormone then diffuses into or is transported into the nucleus. The combination binds at specific points on the DNA strands in the chromosomes, which activates the transcription process of specific genes to form mRNA. The mRNA diffuses into the cytoplasm where it promotes translation process at the ribosome to form new proteins. Example, aldosterone, one of the hormones secreted by the adrenal cortex, enters the cytoplasm of renal tubular cells which contain a specific aldosterone receptor protein. To sum up, we will now discuss the mechanism of steroid hormone action and its effect in detail. The steroid hormone forms a large group of hormone receptor that exerts its effect by a mechanism fundamentally different from that of other hormones. They act in the nucleus to alter gene expression. Steroid hormones, example, estrogen, progesterone and cortisol, too hydrophobic to dissolve readily in the blood, are transported on specific carrier proteins from their point of release to their target tissues. In target cells, these hormones pass through the plasma membrane by simple diffusion and bind to specific receptor proteins in the nucleus, as shown in the figure. Steroid hormone receptors with no bound ligand, that's aporeceptors, often act to suppress the transcription of target genes. Steroid hormones bind with the protein receptors inside the cell rather than in the cell membrane. Within the cell, intracellular receptor ligand complex controls the activities of responsive genes. A large number of nuclear receptors have been identified through sequence similarity to known receptors. Because these hormones are lipid soluble, they readily cross the cell membrane and interact with receptors in the nucleus. Hormone binding triggers changes in the conformation of a receptor protein so that it becomes capable of interacting with specific regulatory sequences in DNA called hormone response elements, thus altering gene expression. The bound receptor hormone complex enhances the expression of specific genes adjacent to hormone response elements with the help of several other proteins essential for transcription, and in this manner either activates or represses transcription of specific genes and formation of messenger RNA as shown in the figure. Once, after minutes, hours or even days, the hormone has entered cell, newly formed proteins appear in the cell and become the controllers of newer altered cellular functions. Many different tissues have identical intracellular hormone receptors, but the genes that the receptor regulates are different in various tissues. An intracellular receptor can activate a gene response only if the appropriate combination of gene regulatory proteins is present, and many of these regulatory proteins are tissue specific. As a result, the responses of different tissues to a hormone are determined not only by the specificity of the receptor but by the genes that the receptor regulates. Example, the specificity of the steroid receptor interaction is exploited in the use of the drug tamoxifen to treat breast cancer. In some types of breast cancer, division of the cancerous cells depends on the continued presence of estrogen. Tamoxifen is an estrogen antagonist. It competes with estrogen for binding to the estrogen receptor, but the tamoxifen receptor complex has little or no effect on gene expression. Consequently, tamoxifen administered after surgery or during chemotherapy for hormone-dependent breast cancer slows or stops the growth of remaining cancerous cells. One more example of steroid analogy is the drug mifepristone. It binds to the progesterone receptor and blocks hormone actions essential for implantation of the fertilized ovum in the uterus and thus functions as a contraceptive. Certain effects of steroids seem to occur too fast to be the result of altered protein synthesis, where the classic mechanism of steroid hormone action through nuclear receptors. Example, the estrogen-mediated dilation of blood vessels is known to be independent of gene transcription or protein synthesis, as is the steroid-induced decrease in cellular cyclic AMP. In conclusion, the effects of steroid hormones provide additional well-studied example of the modulation of eukaryotic regulatory proteins by direct interaction with molecular signals as shown in the figure. Unlike other types of hormones, steroid hormones do not have to bind to plasma membrane receptors. Instead, they can interact with intracellular receptors that are themselves transcription activators. Steroid hormones are too hydrophobic to dissolve readily in the blood. Example, estrogen, progesterone and cortisol travel on specific carrier proteins from their point of release to their target tissues. In the target tissue, the hormone passes through the plasma membrane by simple diffusion. Once inside the cell, the hormones interact with one of the two types of steroid-binding nuclear receptors. In both cases, the hormone receptor complex acts by binding to highly specific DNA sequences called hormone response elements, thereby altering gene expression. Acting at these sites, the receptors act as transcription activators, recruiting co-activators and DNA polymerases with their associated transcription factors to trigger transcription of the gene. The DNA sequences to which hormone receptor complexes bind are similar in length and arrangement but differ in sequence for the various steroid hormones. Each receptor has a consensus hormone response sequence to which the hormone receptor complex binds. The hormone receptors have a highly conserved DNA binding domain with two zinc fingers, and the hormone receptor complex binds to the DNA as a dimer with the zinc finger domain. The ability of a given hormone to act through the hormone receptor complex to alter the expression of a specific gene depends on the exact sequence of the hormone response element, its position related to the gene, and the number of hormone response elements associated with the gene. The ligand binding region of the receptor protein always at the carboxy terminus is quite specific to the particular receptor. With that, we come to the end of today's lecture. Hope you have understood it well. Thank you and goodbye.